have Dr. Hayden. And so for time purposes, his bio is on the app. Dr. Hayden is an accomplished scientist and physician, and I'm very excited to have him. If you would have asked me when I first started my journey who I would have wanted to meet in the HD world more than anyone, it would have been Dr. Michael Hayden. He is my hero. And so just to be able to have worked with him a little bit has been a dream come true for me. But he is here today to talk about Prilenia and proof with us. And so thank you so much for coming on with us today, Dr. Hayden, and I will let you start your talk about Proof HD.
Thank you, Katie, and thank you, Katrina. And what an honor and privilege it is for me to have had this opportunity in the past to interact with you and also to have this opportunity to share some information today. I've been watching a lot this morning and, as usual, been very moved and inspired listening to BJ and Wes and Vicky and Joe and Erica and others as they told their stories. And it also reminds me why Help for HD is so important. So much practical information, so much support that is very unique to this organization, truly providing help for HD. So it's very moving, it's terrific, and I want to congratulate you for doing what you do, which is so profoundly important, very pragmatic. And so today, I want to share with you a little bit on the Proof HD trial, where we are. I've been in this field for over 40 years and deep roots, and these roots have been there to create connections, withstand all the elements. And I was first moved to work in Huntington disease based on my interactions with families in South Africa who really inspired me, imprinted in me early. And Marjorie Guthrie helped me to come to the United States. And so my commitment has always been both to science and advancing therapeutics in particular, but also to the patient organizations. And so this particular drug showed and gave me some hope that there might be some way that we might be able to modify the course of the illness using this drug. And let me share with you why. This is an investigational drug in trials for HD and ALS. Importantly, this is an oral drug, and it's taken twice a day and is very safe and tolerable. And I'll give you some examples from the Proof HD study. And this drug has shown, in fact, is the only drug ever to have shown some maintenance of total functional capacity. This was not the primary endpoint in the study, but it gave us some indication about how to design a proper pivotal phase 3 study. The design of the study was also based on understanding the biology. We now understand that this is a very unique, specific, and selective sigma-1 receptor. And we've seen some effects in terms of maintaining activity up to 5 years. This is in open label, where we know the patients on the drug, and of course, isn't as rigorous as seen in double-blind studies. Of course, as you look at this, the number of patients affected for Huntington disease now thought to be around 100,000. For every affected person, 3 people destined to be ill but not yet ill. Only 2 approved treatments, tetrabenazine, and I took Austedo to approval for the treatment of chorea. And of course, you all know more about the symptoms and signs of what is truly a devastating illness. Now, when I was as head of R&D at Teva, we developed Austedo to treat chorea. And this is what Neurocrine has done also with Ingrezza, to treat chorea. But the families that I interacted with over many years have spoken about the functional capacity, their inability to function, their inability to continue to provide support, to work, to interact with their children and grandchildren. And that's really what we wanted to focus on. This functional capacity goes down even before the diagnosis is made. Diagnosis is really quite late. And families and subjects have emphasized that this is what really is the major burden, leaves them unable to form activities. They become dependent on others for feeding, for eating, for dressing, completing any tasks in daily living. And this was the study that it was an early exploratory trial with numerous caveats, but the important point was, based on analysis, the placebo went down as expected, but patients who'd been on this, subjects on predopidine, did not show there was a very significant difference relative to placebo in terms of their ability to continue to function. So you gave them a year, at least a year, because this was based in 52 weeks, more ability to stay employed, more ability to take care of themselves, clean the home, continue working, and do things that matter to enhance the quality of life. We also knew at the same time that this drug was actually binding a particular receptor in the brain. And this was using imaging studies of the brain, where you give something that labels the receptor you want. This is a sigma-1 receptor binder that labels sigma-1, and then you give your drug, and you see, previously you give your drug, before that experiment again, and you see how much you can block that. And if you block it, that means that your drug is binding that particular receptor. And here you can see, when you give the drug and you then treat with fluspidine as well, you lose the signal, which means the door that opens that receptor is blocked by your drug. It tells you that your drug binds sigma-1. Now we know, and we've shown in a whole host of studies, around 40 independent studies with 40 investigators around the world, that if you can activate this drug, it's neuroprotective. It does this by reducing stress, by reducing inflammation, by clearing toxic proteins, by enhancing energy production, remarkably increasing the connections in the brain, and also enhances secretion of protective factors, like one important one, which is BDNF. So we know that. We also know that it's acting through sigma-1, because genetically you can delete a single gene in a mouse, for example, and when you delete one gene out of 20,000, you actually see that all the effects are lost. It's acting through the sigma-1 receptor in those experiments. And here's just one experiment. This was done by Chris Ross's lab, where you take cells from the brain, from HD neurons, from an animal model, and you can see, you just look at baseline, cell death is increased, and then you treat with pridopidine and you can actually reduce cell death. And this is again shown to be directly mediated through the sigma-1 receptor. So this is important data. Now, importantly too, it's not just about the receptor and the mechanism of action, but a drug that's going to be used for Huntington disease needs to be safe and tolerable and easily administered. Otherwise, people won't have access. And we have a lot of data now, and I'll show you some new data, just showing that this side effect profile is comparable to placebo. No serious adverse events related to the drug, and we've got a lot of experience. And so this is also something that's very helpful, taken orally, safe and tolerable. So for example, these are some of the side effects that we've seen from an integrated list. And what you can see, placebo and the treated group really have the same numbers. There's no real difference between them, nothing significant, not only in terms of serious adverse events where none are attributable to the drug, any side effects. So this is really also encouraging that this is so safe and tolerable. Now, sadly, at the moment, Prilenia and pridopidine is the only drug in later stages of clinical development for HD. This came from a BioCentury, which is a biotech sort of review and important document and publication. And here you can see a whole host of others that are here. Sage, that lovely presentation just by colleagues at Sage now. But all of them, this is the only drug currently in phase 3. And when you develop drugs for any indication, you have to go through phase 1, which is really about safety, phase 2, giving you some proof of concept, allowing you to plan your phase 3 study. And that's where we are right now, sadly alone. Now, important to know that even though you're in phase 3, it's not certain that you're going to have success. And it's just important to maintain the balance. So if you look in the literature for rare diseases, and neurology is more or less the same, you can see there's a 60% chance of success that this will proceed to a regulatory submission. And another way of saying it, there's a 40% chance that there won't be a regulatory submission for whatever reason. Sometimes it's safety, sometimes it's efficacy, sometimes it's other factors that play a role. So it's important to know that even though we're in phase 3, it's not certain that the study will be a success. However, we believe that there's enough data to be significantly hopeful about this study. And so this study was enrolled during COVID. It was designed and powered to replicate data from prior trials that showed a functional and beneficial effect on functional capacity. And the trial enrolled the first patient in Kansas in October 2020. We have the studies going on in Europe as well as North America. In North America, Andrew Fagan, who's the outgoing chair of the HSG, Sandra Kostig, who's the incoming chair of the HSG co-chair. In Europe, it's Dr. Ralph Freeman and Anne Rosser, who's the chair of the European Huntington Disease Network. And the study, based on its possibility of success, has been formally endorsed by the European Huntington Disease Network. So this is a phase 3. There's one group that's on the placebo, one group we know the right dose on 45 milligrams. We aim to enroll 480, but in fact, there was such a waiting list that we enrolled 499, just about 500 patients over. And also the study was enrolled 2 months ahead of schedule during COVID. So this talks to the commitment of the community, this community that has been so responsive to this particular possibility. Our primary objective is to look at the effect of pridopatine on functional decline using a regulatory accepted endpoint. And both the FDA and EMA have told us that this is sufficient as a single primary study, pivotal study for approval if successful. And now we're into the open label. The open label means that those people who are on placebo now go on active drug. Everybody receives pridopatine, and we've had 100% conversion into the open label at this point. The study is going on in 29 sites in 9 countries in Europe and the United Kingdom. So really all over the world. The most east is Poland. And then when we look here in North America, 30 sites, 25 in the US and 5 in Canada. And this is really the current data. So we've had a dropout in this study, people of just over 5%, very little in 22 months, very unusual. We had powered the study for a dropout rate of 20%, which would be more likely to be seen. So very low, again, ahead of schedule, October 20, been no safety signals of concern. And some of the patients have now accepted and completed 65 weeks and gone into open label. So again, no serious adverse events related to the drug at this particular point. The study is ongoing. We're just about 9 months away from expected results here, end of Q1, early Q2 in 2023. And after that, we will know whether we've been able to show that this orally taken, safe and tolerable drug may be effective in slowing progression as measured by total functional capacity. So this is an exciting time for us. There's also a study, Mass General and Harvard Medical School asked, and we were in a competition, and they're also funding an ALS study that's ongoing through the first platform trial in ALS, where there's 4 drugs being tested at the same time. We're also currently planning a study in people destined to be ill, but not yet ill, but close to onset. And that study would start in 23 for prodromal HD based on various biomarkers and other clinical signs that are not sufficient to make the diagnosis, but give people indications that that onset is going to be within the near future. We're also continuing to talk to our colleagues. And I think it was Erica talking about juvenile Huntington disease. We're, of course, very interested in that community and want to make drugs available for that community, and also considering planning a study in juvenile HD. Important to note that the capsule that's given is really given for people over the age of 16. We're now looking at smaller, we have capsules that are smaller, 22.5 milligram, that could be used. But when you get to children that are in the ages of 2 to 5 or 2 to 6, you need a different formulation. and we're busy working on creating a formulation. But the initial trial in juvenile HD would follow the results, and then we would plan that for some time in 2023. So let me just say, I'm deeply moved by this community. I've been imprinted 40 years ago by the courage, the resilience, and today is no different. What this conference has done with Help for HD has deeply shown the commitment of partners to their partners who are suffering deeply. And we're also very grateful that the Huntington's community and study participants and families gave us their trust and continued to collaborate with us in this particular study that's ongoing. So we hope and pray that this will be successful. It's not a sure thing. We're looking forward to getting those results in the near future. And of course, we'd share them with you as we go forward with the results in about 9 months from now. So thank you all. Congratulations again to Help for HD and Katie and Katrina. Amazing to see what you're doing and providing a place for the voice of families and people in different ages, different domains, talking about the experience. I know how comforting that must be for everybody. So thank you all. Thank you for your attention.
Thank you, Dr. Hayden, and thank you for sticking with us for 40 years. Can you imagine 40 years of working in HD, what you have seen? Unbelievable.
Yeah, and that's right. And I know you're thinking, you must have started this when you were 8. No, I was a little older.
Oh, thank you so much.